Hello, everyone. Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. This is the last seminar of the season, and I think we'll wrap up the series with some really excellent talks today. My name is Kate Criswell, and I'm a postdoc at the University of Cambridge, and I will be moderating today with Whitney Edwards, a postdoc at UNC Chapel Hill. We are excited to highlight the work of our outstanding postdoctoral members. Today, Margherita Perillo from Brown University and Hector Sanchez-Iranzo from Karlsruhe Institute of Technology will share their research. Each speaker will give a 20-minute talk, followed by approximately 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box at any point during the talk, and the moderators will read them out once the talk is finished. The moderators will also give a verbal warning to the speakers when there's 1 minute left in the talk time. And now I'm very happy to introduce Margherita Perillo. Margherita grew up in Italy and did her PhD at Stazione Zoologica Anton Dorn in Naples, Italy, through the Open University of London, where she studied the evolutionary origin and diversification of pancreatic cell types during sea urchin gut morphogenesis with Maria Ina Arnone. She is currently based at Brown University, working with Gary Wessel on cellular mechanisms of organogenesis and primordial germ cell migration. Today, Margherita will tell us about her fascinating work in the sea star, focusing on cellular mechanisms driving tubulogenesis, insights from the sea star larva. I am very excited to hear more about sea star development today, so Margherita, go ahead whenever you are ready.
All right, Kate, can you hear me?
Sounds good.
Yeah, you can hear me. Thank you. All right. Thank you, Kate, for your introduction. And also, I would like to thank you, SDB, for selecting my abstract to give a talk. Selecting my abstract to give a talk today. And so I'm a developmental biologist, and I work with this beautiful animal that you can see on the slide. It is the sea star Patira miniata. And so I'm deeply fascinated by how, in general, individual cells can group together to form tissues, and how this tissue can form more complex three-dimensional structures like organs. And I'm going to tell you more about these organs depicted in this cartoon during my talk. And so there are several factors that act on the individual cells within a growing organ. For instance, the intrinsic factors like the cell mechanics, and extrinsic factors like the guidance coming from the microenvironment. And last, there are some cell types that need to migrate in order to get their final position within the growing organ. And so all these factors are really critical for the proper tissue function and 3D patterning, that if it's altered, it's going to cause major embryonic malformations like a loss of organ symmetry and function, or in the adult body can cause defects in tissue regeneration. And so to tackle this question of how individual cells group together to form these distinct organs, during my postdoc, I decided to subdivide this in two specific questions. The first one being what drives the formation of tubular organs, and the second one being how do cells migrate together to reach their position within this organ. And then for the sake of time today, I'm only going to talk about this first project. So what drives the formation of tubular organs? And this leads me to define what tubular organs are. So these are organs with a very important function of carrying, transporting liquids, gases, cells, or nutrients. And I also want to add that the tubulogenesis, that is the formation of this hollow tubular structures, is also a very important first step in the organogenesis of many organs that will eventually have a different shape. And so what we know about this process largely derived from select model system. Like for instance, I'm showing you here the fly trachea, the fly salivary gland, the zebrafish inner here, or the mouse embryonic lungs. And so all these systems, despite the fact that they share common features, they do develop very differently. So for this reason, no single model system can fully represent tubulogenesis 100%. And so if we want to understand, if we want to define the fundamental steps in making a tube, I think it's very important that we start to study more tubular organs and also organs that are simpler. And so the system that I show you on the slide, they belong only to two groups of animals. So there are other protostomes here, or there are chordates. Chordates being the group of animals that include tunicates and vertebrates. And so to fill this gap here, I decided to establish an another model, so an early branch deuterostome model, to know more about this process of tubulogenesis. And so these animals have an important phylogenetic position that from a biomedical perspective, but in addition to this, they also have a series of advantageous experimental approaches that we can use. And so among the canines, I chose to develop specifically the sea stars because the larvae of these animals develop two long tubes that are very interesting for this purpose. And so to summarize the work of many people, including mine, I'm now going to show you the life cycle of a sea star with these cartoons. So the eggs are fertilized in the ocean, and the larvae grow and live in the ocean. And now with the magenta color, I'm going to highlight where the sea star tubes are. And I use this word for sake of simplicity, of course. And so the precursor of the tubes, they start to appear during gastrulation as these mesodermal precursors at the tip of the growing gut in the gastrula. Then during the larva stages, from the tip of the growing gut, you can see two bilateral tubes that grow, elongate, and extend towards the posterior side of the larva. And then eventually in the later larva, these two left and right tubes will merge together to form a closed structure that is, let's say, vascular-like. So the stage of late larva that you see here, now I'll show you how it looks like in a real larva. So this is a sea star larva where I lighted in a dotted magenta line where the tubular organ is. And these arrows that you see popping here and there are pointing at sites along the tube where twitching, contraction is happening because there are muscles around the structure. And the structure is vital because it's a buoyancy tank. So the structure allows for the larvae to balance in the water column. And just because it is nice, I'll show you here in the stomach, you see these things moving. These are actually algae that is a favorite food of sea star larvae. And so why do we like this system so much? It's really trackable. So as you can see from this video, these larvae are optically clear. And so basically I can grow them in a 6-way plate like an organoid, but I can study the organogenesis live inside an intact organism. So we don't need to dissect anything. Everything happens within the organism. Also, we have a very useful model organism database for this species. And we have a series of genetic tools that we can inject in oocytes, as you can see from this video here. And so with this system in hand, now I can ask how tubulogenesis works. And I'm going to focus today on two factors, intrinsic and extrinsic. Starting with the question, what are the mechanisms within the cellular mechanism within a growing tubular organs? And so I realized that if I wanted to look at cellular dynamics during tubulogenesis, I needed a way to look at this while it's happening live. So for this reason, during my postdoc, I developed ways to immobilize and live image sea star larvae for a very long time. In fact, as you can see the scale of this video here, these are hours. And so you're looking now at a sea star larva where the actin is magenta and the nuclei are marked in green. And you can, I hope you can clearly see that thanks to video like this, I'm able to see individual cells moving within these growing tubes. And I apologize for this video being a little bit chunky on your screen. It doesn't play the way it plays on mine, but I guess you get the concept. And so analyzing many videos like this, I was able to track individual cells while they're growing along the tube. And I can do this on two different directions. First on the anterior-posterior direction, and second on the left-right direction. And so, for instance, if we plot individual cells movements along the anterior-posterior axis, what we can see is that there are two main stages. So in the first stage here, there is a sharp movement of all these cells towards the posterior side of the larvae. And this is more or less 8 hours in average. And then for the other 20 to 30 hours, the cells don't move along the anterior-posterior axis anymore. They kind of stay where they are. So if we look at the total displacement, so how much individual cells move during these two stages, we see that the majority of the movements are accomplished in the first stage, so in these 8 hours. But then if instead we look at the left-right displacement, what we see is that the majority of this movement on this axis happens instead on the second stage that is longer. And so this suggests us that in order to form this tube, there are two kinds of movements. First, there are movements towards the posterior side of the larva that allows for tube extension. And then there is a second kind of movements that allow for tube expansion. And so in many movies like this, I realized that while these cells are migrating, they are also dividing. So I can see mitosis, as you can see in this video. And so I next ask, when and where does cell division happen during tube elongation? And so to get bigger numbers, I perform an EDU experiments where an EDU molecule gets incorporated during S phase in cells, and then it's going to mark cells that were proliferating. And so I did this at different stages, and I summarized the results of this experiment in this heat map here. And what I found is that the majority of cell division, so the majority of cell proliferation, was happening at the first larva stage. And in particular, I saw that proliferation was particularly high in two proliferated zones, that is in the middle and in the tip of the growing tubes. Then I asked another question. So since there are two tubes at this stage, L1, so there's a left tube and a right tube, I asked whether there was a difference proliferation between these two structures. So what I found is that the only difference between left and right tube is that there is increased cell proliferation at the tip of the left tube, exactly here. So this area of the tip of the left tube is actually where the tubular organ gets touches the ectoderm and creates an opening towards the outside environment that is called the hydropore canal. So if you look at this view of the hydropore canal, you can see that this magenta color shows the cell proliferation happening. And so after having defined actually cell division is important, it's happening, during tubulogenesis, I then ask whether cell cycle progression was required for tube initiation and elongation. And so to this aim, I use a drug that arrests the cells in G1 and so prevents cells from going in S phase. And I had these drugs at two different time points. The first time point being at the onset of tube formation. So the question was, is cell division, is cell progression required in order for the tube to form in the first place? And then to test whether the experiment was working or not, I perform again an EDU experiment, because if the drug is working, we shouldn't see any EDU incorporation since there is no S phase. So what I found here is that first of all, the drug is working, there was no cell proliferation happening. And second of all, the tube was really, basically didn't form, as you can see also in this picture. And so then I ask a second question. So once the tube is already formed, do we need, does it need cell cycle progression in order to keep elongating? And so I had this same drug to arrest cells in G1 at a later time point, once the tubes are already formed. And what I see also in this case, again, is that cell proliferation is blocked, and then the tube doesn't grow anymore. So basically the tubes keep the same length that they had when I added the drug to arrest cells in G1. And so this cell progression, cell cycle progression is fundamental to start and then to keep elongating. And so what I told you so far is that during tubulogenesis, there are two stages of cellular dynamics that allow for tissue extension and expansion. And then, so the cells are able to migrate and divide at the same time. And this cell cycle progression, this cell division is really critical for the tube, not just to initiate, but also to elongate. And this marks a very important point during the evolution of tubular organs. So I'm now moving to the second factors that affect the behaviors of cells within a growing tissue, that are extrinsic factors. That means I look for embryonic pathways that are known to be important for the embryos, and I ask how these pathways specifically affect the morphology and the formation of these tubes. And so for sake of time, I'm going to only show today two of these pathways here. And I start with the Wnt pathway. So to really summarize a lot of how the Wnt pathway works, the only thing you need to know for my slide here is that there are a series of Wnt secreted ligands, and they can bind a series of freezer receptors on the target cell. And this is going to activate beta-catenin and then can enter the nucleus. And so I first use a drug to block the secretion of all the Wnt ligands. And so what I see is that while in a control larva, you see the tube grow anteriorly towards the posterior side of the larva, if you can follow this magenta arrow. When instead we block the secretion of all the Wnt ligands, what happens is the tube, instead of growing posteriorly, it grows dorsally, if you look at these other arrows here. And so this tells us that the Wnt are important for the proper orientation of the tubes in the embryo. And so there is one of the freezer receptors that is specifically expressed by the tube cells, as you can see here. And with a CRISPR-Cas9 approach, I knock out this gene, this freezer receptor. And what I see, if you look at the cartoon here, that we recapitulate the same issue that happened with the drug. So instead of growing anterior to posteriorly, the tube grows toward the dorsal side. And this is something that I quantify by measuring the angle that the tube form with anterior posterior axis of the embryo. Okay, now I move to the second signaling pathways that work on, that is the FGF pathway that is important for the branching of many tubular organs also. And so the FGF receptor is expressed in selected tube cells of the C-star larvae that you can see here. And again, when I knock out these FGF receptors, what I see is that the tubes do not form at all. So you see they look like extremely small, almost don't form. But the FGF pathway is well studied, and there are a series of drugs that one can use to stop this pathway at different levels. For instance, there is a drug that blocks the kinase domain of FGF receptor, or there is another drug that blocks a kinase that is downstream of FGF receptor. And so to have a more solid data, I use this drug. And what I found is that if we stop the FGF pathway at different levels, we always get the same results where the tube do not form, as you can see here. And also this is something I can quantify if I measure the area of the tubes. And one of the downstream activator then of the FGF signaling is ERK. So ERK can be activated when it's phosphorylated. So I look for the expression of phospho-ERK, so active form of ERK in the C-star tubes. And what I found is that phospho-ERK is in the nucleus of the tube cells, mostly the ones at the anterior side. And when we knock out FGF receptors, this expression disappears. So it is indeed phospho-ERK dependent on the FGF receptor. And so since phospho-ERK is important for its activate cell proliferation, and then checked whether this was also the case for the FGF receptor. So when in a control larva you see here, in magenta, you see a lot of cell proliferation, as I showed earlier, happening in the tubes. When I knocked out FGF receptor, you can see that the cell proliferation in the tube is minimal, and this is something again I can quantify. And this decrease in cell proliferation is very specific to the tube cells, because if you look at the esophagus that is here inside, you can see the bottom controls and the FGF receptor knockout, cells keep proliferating. And so if you look now at control larva, again, the tubes are highlighted here in magenta. And then I show you a larva that is knocked out for an FGF receptor. You can see that the gross morphology of the larva really looks the same. They are both digesting, they really look alike. The only difference is that the tubes are completely missing. And so this tells me that now I have a tool for to genetically or chemically ablate the tube of the C-star larva. And I did use this tool in order to perform a differentially RNA-seq experiment of control larvae compared to larvae where the FGF receptor was blocked. And thanks to this approach, I was able to find a series of candidate genes that I found to be then expressed by the tube cells. And then just to give you an example, one gene that I found, I'll show you 612. This is a transcriptional factor that is, as you can see here, is expressed by the tip of the left tube. So again, here, this gene is expressed in the hydropore canal, the structure that is opening towards the outside environment. And when we knock out 612, what happens is that this hydropore canal, as you can see here, doesn't form anymore. You can also see this in the cartoon. And so this tells us that there are a series of different cell types in these tubes, and then we can identify specific regions that are under the constraints of very specific transitional factors. And so now I'm going to summarize what I tell you so far. So I show you that during topogenesis in these C-star tubes, there are two kinds of movements. So cells can migrate first toward the posterior side and then on the left-right axis. And while they're migrating, they can also divide. And cell division is fundamental for the tubes to form. So I'm going to show you a series of signal pathway important for embryogenesis. Also act specifically on the tubes for the growth and for the orientation within the growing embryo. And last, I just mentioned that I found a series of candidates that tells me that there is a great diversity of cell types. So I learned something about the tissue patterning in this case. And the fact that there are many cell types within these tubes was also confirmed by a recent paper that came out in bioarchive from the Hinman lab, where they use a single cell approach to find that already at early stages before the tubes are formed, they were able to identify subtypes within the tube cells. So this was really great. All right, and so to finish my talk, I just want to say that this major theme of organ development and evolution is going to also be the main focus and the foundation of my future independent research program at MBL that very kindly offered me this opportunity of working with them. And I have to say that for a developmental biologist who works with marine animals, working at MBL is definitely like a dream job.
1 minute left.
Okay, thank you, Kate. Okay, and now before I move to the acknowledgement session, just want to share one slide with you, because as a scientist, there's also something besides science that I really care about, that is a conversation on how to improve the culture in academia. So the issue is that for too long, too many labs were managed like a feudal system, where basically the lab PI was perpetuating a culture of harassment that is perceived still by many trainees and many PI also that like normal in academia. So something that you have to cope with if you want to stay in academia. In fact, many brilliant scientists unfortunately left academia because they thought they couldn't handle it anymore. And so the good news here is that there are a lot of PIs that say that brilliant people who are actually writing very impactful papers in science and in Nature, where they actually point out the problem of harassment in academia, bullying in academia, and they also give solutions both to the university and to the funding agencies. And I really suggest everybody to read these articles. And last, I try to give my very small contribution to this conversation by writing a few blog articles about what bullying in academia is and how people can handle it and what are the kind of solutions they can find. And so this the take-home message I want to give you with these slides, it's simple, that it's that there is a bright future, I think, for science, because we are finally talking about the problem we have. And I really think that the more we talk about the issues we have in our community, the more we will find ways to solve these issues all together with more ideas. And so now I just want to move to acknowledgements. I want to thank the Western Lab for allowing me to work on organogenesis in a lab that instead focuses on reproductive biology. And then I also would like to thank all the people on the slide who are collaborators or mentors that I had during my postdoc. And very last, I just want to say that together with my colleague, Rossella Annunziata at Sezione Zoologica Tondor, we are co-editors of a special issue on marine animal organisms. So if you have any paper where you cellular or developmental biologists working with marine organisms, please, submissions are well accepted. And so thank you everybody for listening to me.
Thank you so much, Margherita. We have a few questions. The first is from Viraj Dorehal. Beautiful images and videos. I'm wondering about the function of Wnt signaling in patterning the tube formation. Why do you think that in the absence of Wnt signaling, the tube grows dorsally? Is it because changes in the body axis of the larva?
Yeah, okay, thank you for this question. So that's really interesting. It's something I'm gonna investigate more in the future. So I think that here what happens is that the non-canonical Wnt pathway is working. So my hypothesis is that it's a specific through the FRITZ-127 receptor, the orientation of the cell division of the tube cells is changing. And this is, and because of this, the cells are going toward, the tube itself is going towards a different direction. That's my hypothesis, but I need to do the experiment to actually test if this is true or not.
Okay, we've got another question from Michelle Stars-Guyano. Cool model system and great talk. Is the lumen present for the whole time you are examining the outgrowth and migration of the tube cells? And are the migrating cells moving in a monolayer or through a multi-layer in that time?
All right, thank you, Michelle. This is also a wonderful question. So this is, first of all, let's start from your second question. This is a monolayers of cells. And this is not a de novo formation of the lumen. So the lumen is already formed while the cells are migrating. So the cells are coming from the tip of the growing gut. So the moment they start to migrate towards the posterior side, they already have this conformation of apical basal polarity with the apical side facing the lumen inside the structure.
Okay, one more question. This is from Marsha Lucas. Have you tried overexpressing 6, 1, and 2? Do you see the right tube mimicking the turn on the left tube?
That's a fantastic question. Thank you, Marsha. I love it. No, I haven't tried this yet, but I think it would be really, really cool to try. Thank you. You gave me a very good suggestion.
I've got a quick question as well. I was wondering, you mentioned about how having to immobilize them for doing all of this beautiful live imaging. Could you talk just a little bit about the sort of technique you developed to do that?
Sure. So I basically always admire the beautiful live imaging that the zebrafish community can do. And so I basically use a very similar protocol with the low-melting agarose. The difference is that since these larvae are way smaller than a zebrafish larvae, it's more complicated to keep them in between slides, but this is what I do. I really follow a very similar protocol to zebrafish.
Great, thank you. And we've got one more question from Zainab Afzal. Really neat system. Have you tried increasing cell division? I'm wondering if that would just make a longer tube or if there are mechanisms to ensure cell division is happening only at a specific time as the tube grows.
I wouldn't know how to increase cell division, but this would be interesting to try. I didn't try it. I would need to figure out how to perform this, but thank you for the suggestion.
And then one more has come in from Pericles Paganos. Super cool project and talk. Any ideas on which cell types provide the FGF ligand that cross-talk with the tube cells?
Also, this is a great question. I don't know which of the cells of the tubes these are, but I really think that thanks to the single cell RNA-seq that other labs are doing, I will be able to find a lot of good candidates there. So thank you.
Great. Any other questions? If not, thank you so much, Margherita, for a beautiful talk. And I think we can go ahead and move on to our second speaker.
Hey, hello, everyone. It is my great pleasure to introduce Hector Sanchez-Iranzo. Hector studied pharmacy and biochemistry at the University of Valencia. He then moved to Madrid, where he did his PhD at the Spanish National Center for Cardiovascular Research in the group of Nadia Mercade, where he published multiple papers on the mechanisms of heart regeneration in zebrafish. In 2018, he joined the lab of Alba de Munoz at the European Molecular Biology Laboratory for his postdoc, where his work focused on the generation of notch patterns in zebrafish notochord development. And since 2020, Hector has started a transition to independence position at Karlsruhe Institute of Technology, where he is studying how gene expression is coordinated with mechanics during notochord development. And I'm very excited to hear your work on the notochord today, Hector, and you can take it away whenever you're ready.
So thank you very much for the introduction, and I'm very happy to participate in this wonderful seminar series. So my research interest is understanding how biological systems self-organize to form the complex structures that we see across the animal kingdom. So you can see, for example, these developing zebrafish where somehow cells coordinate between them to determine their differentiation and also their location and shape. And ultimately, if you think about it, most of the information to build an organism is in their genome. So in our DNA, we have the sequences that code for proteins, but also the sequences that determine when and where these proteins are going to be produced. Then we have some proteins that can regulate other proteins, the expression of other proteins. And when we have several proteins that interact with each other, they can form complex gene regulatory networks. During development, cells use these gene regulatory networks to determine and to generate patterns of gene expression. These patterns of gene expression ultimately determine cell fate and also the shape and location of the cells. And this is a mechanical mechanical process that involves the generation of forces. And during this talk, I will focus mainly on this first part, but also keeping in mind always like the whole picture. Well, so to understand how patterns are generated in the notochord, I use the zebrafish notochord model. So the notochord is this organ that you can see here, for example, in the zebrafish, and extends from the anterior to the posterior end of the embryo. And it constitutes the structural axis of the embryo. When the notochord is fully developed, it resembles almost like a perfect cylinder, as you can see in this movie. And it is formed by two different cell types, the vacuolated cells in the center of the cylinder and the sheath cells that cover the surface. The vacuolated cells generate a huge amount of pressure inside the notochord, and the sheath cells produce extracellular matrix that withstand this pressure. So overall, the notochord behaves something like a cylindrical balloon that is inflated, and this gives some structural support to the embryo. But what I found very surprising is that these two different cell types that have so different functions and shapes, both of them are derived from a common pool of progenitors. They are these coin-shaped cells that are perfectly aligned one after each other, just like a stack of coins. And then some of those cells will become vacuolated cells, and some of these other cells will become sheath cells. We can visualize this process in vivo. Here you can see, for example, how some cells start to grow the vacuole, and then the neighboring cells starts to move to the periphery, to one side or the other, until we have the vacuolated and sheath cells that form the mature notochord. Also importantly, this process occurs from the anterior to the posterior. So the cells that are in the more anterior end of the embryo, they are more developed than the ones that are more in the posterior end. So the question now is, how is cell fate determined in the notochord? Well, so when I started working on this, there was very little information about it. So it was only known that if you overexpress the notch pathway, then all the cells become sheath cells. And in contrast, if you inhibit notch, all the cells become vacuolated cells. It was also described that the main ligands in the notochord are JAGET1A and JAGET1B. And interestingly, when the human homologs of these ligands, that is JAGET1 or the notch receptor notch2, are mutated in humans in the Alagille syndrome, then it leads to vertebrae malformation, highlighting the importance of studying the notochord in zebrafish and how it can explain phenotypes in humans. So, notch signaling is a master regulator of cell fate. So it can regulate cell fate in almost every organ. And just to cite some examples, we have the retina, heart, or pancreas. And something particular about the notch pathway is that it mediates signaling by direct cell-cell contact. So if we model this kind of signaling, then it has been shown that notch signaling can generate a great diversity of patterns. And as I was commenting before, so the main components of the notch pathway are the notch ligand and also the notch receptor. And it has been shown that depending on the ligand, notch signaling can generate different patterns. And for JAGET1, that is the ligand in the notochord, what has been described is that it always generates lateral induction patterns. So let me explain how this lateral induction would work. So we have the ligand that binds to the NOTS receptor in the neighboring cell, and then the intracellular domain of the NOTS receptor is cleaved and translocated to the nucleus, where it can activate the expression of the ligand. This ligand then translocates to the surface, where it can in turn, again, activate NOT signaling in the initial cell, but also in the neighbors. And this generates a positive feedback loop, where in the end, all the cells are both positive for ligand and for NOTS. We can model this kind of pathway using differential equations. And if you have just one single cell that is ligand positive, then this pattern would spread like a wave until all the cells are both positive for NOTS, ligand, and receptor and signaling. So, and then also all the cells would be completely homogeneous. So then I wanted to visualize the pattern in the notochord, and to do this, I generated a back reporter, jagged 1A scarlet, and I crossed it to the NOTS activity reporter, TP1GFP. But surprisingly, what I found is that this pattern is far from homogeneous. Instead, it's really alternating between cells that are expressing the ligand and cells that are activating NOTS. Well, so this pattern resembles much more to the lateral inhibition that has been described for other ligands, such as JAG2 or delta, but it has never been reported for JAG1. And in this lateral inhibition, when the ligand binds to the NOTS receptor again translocates to the nucleus, but in this case, it activates a repressor that inhibits the expression of the ligand. So if you remember, before, NOT signaling activates the expression of the ligand, but here in lateral inhibition, NOT signaling inhibits the expression of the ligand through the expression of a repressor. And this pathway generates B-stable states where every cell should decide whether to become receiver or sender. This kind of patterning can also be modeled. And if we do the modeling in the cellular arrangement of the notochord, what we can see is that generates an alternating pattern that resembles much more to what we see in the notochord, for supporting that what we have is a lateral inhibition patterning. Well, so a key feature of lateral inhibition is that when you have one cell that is expressing the ligand, it will inhibit the expression of the ligand in the neighboring cells. And here we have an image similar to the ones before, where we have the ligand in magenta and NOTS activity in green. And what we can see here is that in general, it's very difficult to find two neighboring cells that are JAG1 positive. So then I wanted to test if NOT signaling is mediating this lateral inhibition. So then to do this, I inhibited NOT signaling. And I did this by using the JAG1A and JAG1B morpholinos. So these morpholinos has been previously validated, and importantly, they can inhibit the endogenous JAG1A and JAG1B genes, but they cannot affect my reporter JAG1A in scarlet, allowing me to visualize still the patterns. So when we inhibit, when we use the morpholinos, the first thing that we observe is that specifically we eliminate NOT signaling, so the green signal, in the notochord, but not in the neighboring tissues, because they would be activated by other ligands. And then the other striking thing is that if we inhibit NOT signaling in this way, then we can see a lot of JAG1 cells that have neighboring JAG1 cells, as you can also see in this quantification. So concluding that inhibition of NOT signaling disrupts lateral inhibition patterning in the notochord. Well, so a key component of a lateral inhibition network is the repressor. So which is the identity of the repressor in this network? To identify it, I took advantage of some single cell RNA-seq data that was previously published, and I reanalyzed it focusing on the notochord cells at the stages that are relevant for notochord patterning, that are 18 hours and 24 hours per fertilization. And I focus on the genes of the family of HER and HES that are known to be NOTS targets and also transcriptional repressors. And I found that HER6 and HER9 are the most highly expressed. So if these genes are repressing JAG1A expression, then they should have a pattern that is alternating or complementary to that of JAG1. And to visualize this, I did this hybridization chain reaction to visualize the mRNA of all these genes. And what we can see is that JAG1 signal is something like in the gaps where there is no HER9 or HER6. So this pattern is compatible with HER6 and HER9 being repressing JAG1A expression. Well, but then do they really inhibit JAG1A expression? So to test this, I used the JAG1A M-Scarlet reporter that I showed you previously. But now what I did is overexpressing GFP P2A HER6 in some random cells in the notochord. Then, so the cells that you see here in green, they are also overexpressing HER6. Then I did some segmentation and I looked at the intensity of JAG1A reporter inside the HER6 cells, and I compared it to the levels of JAG1A outside of them. And what I could see is that when I overexpress HER6 or HER9, then the levels of JAG1A are lower than when we just express GFP as a control. Further, so concluding that both HER6 and HER9 are the repressors of JAG1A in this network. Well, but now, until now, I have explained how this pattern is generated, but does it matter for fate? So first, I use the JAG1A M-Scarlet and TP1 reporter to visualize in vivo the fate of those cells. And what I could see is that it's very clear that JAG1A cells become vacuolated cells and TP1 cells become sheet cells. Showing that these are the first early markers of cell fate in the notochord. But well, but this is only correlation. So are they really sufficient? Are the components of Notch pathway sufficient to determine cell fate in the notochord? So first, I tested the role of HER6 and HER9. And what I did is I inject HER6 and HER9 constructs together with GFP as before, and then I let the notochord to develop. First, if I do this just with GFP, then I can see that GFP positive cells can become both vacuolated or sheet cells. But then if I overexpress GFP HER6 or GFP HER9, most of the cells become sheet cells, indicating that HER6 and HER9 are sufficient to determine sheet cell fate. Then I wanted to focus on the other main component of the pathway, that is the JAG1A ligand. So does it have any role in determining fate? And when I did the experiment, similar as previously, what I found is that surprisingly, JAG1A positive cells tend to become vacuolated cells. And this result was surprising to me because in the end, if you think about it, when you are overexpressing a ligand, I would expect an effect in the neighboring cells, not in their own cell where I'm overexpressing the ligand. So what's the mechanism of this? How can the ligand have an effect on fate on its own cell? So, well, first, I thought about the possibility of a role of JAG1A intracellular domain. So it has been shown that when Notch signaling, the Notch ligand and the Notch receptor interact, not only the Notch intracellular domain is cleaved, but also the intracellular domain of the ligand is also cleaved and translocated to the nucleus. And what it has been suggested is that this intracellular domain of the ligand can then inhibit Notch signaling and produce some reverse signaling. So if this hypothesis is correct, that there is a role of the JAG1 intracellular domain, the JAG1 intracellular domain alone should be able to promote vacuolated cell fate. However, when I did the experiment, I found no role for this JAG1 intracellular domain. So then I thought a little more about the network and how it could be possible that the ligand could promote vacuolated cell fate in its own cell. So this is the network I was describing before, where the ligand interacts with the receptor through transactivation, and then the receptor activates HER6/HER9 in the neighboring cell. Then this HER6/HER9 would inhibit the expression of the ligand, and as there is very little ligand, the receptor in the original cell would not be active, and HER6/HER9 would not be active, and sheet cell fate will not occur. Thus, if you look at this, everything is activatory except for this, that is a single inhibitory signal. So then ligand would inhibit sheet cell fate, promoting vacuolated cell fate. But there is a second possibility, and it has also been described that when the ligand binds to the receptor, and they are in the, well, when the ligand and the receptor are in the same cell, they can mutually inhibit through a process known as cis inhibition. So I want to test the role of these two different pathways into the two different circuits in determining cell fate. So to do this, first I overexpress JAG1A in some cells, and I evaluated the level of HER6 and HER9 in its own cell. And what I could see is that, in fact, HER6 and HER9 levels are lower in the cells where I overexpress JAG1A. However, we still cannot know if it is occurring through this circuit or through this cis circuit. To distinguish between them, then I evaluated the expression of HER6 and HER9 in the neighboring cells. And what I could find is that HER6 and HER9 was, HER6 was not induced in the neighboring cell, and HER9 was induced, but to a limited extent in that neighboring cell. So this suggests that the main circuit is probably the cis circuit through this process of cis inhibition. But then it's like, I wonder, why is it important, or is it, why there is this cis inhibition that is stronger than trans interactions? Is it relevant? Does it make sense? Why is it important? So then I did some mathematical modeling. And in this modeling, I used different values of trans interactions and different values of cis interactions. And here you can see the results of the modeling, where in the x-axis, I saw the levels of the strength of trans interactions, and in the y-axis, the strength of cis interactions. Here in the colors, you can see that red is the region, are the values where you can see patterning, and in blue, you can see the values where there is no patterning. And surprisingly, all the values that lead to patterning are in this side of the diagonal, where cis interactions are stronger than trans interactions. So concluding that, in fact, having a stronger cis and trans interactions is something needed for patterning. So in conclusion, I have shown that JAG1A generates a lateral emission pattern, and that HER6 and HER9 are the repressors in this network, and that pattern generation requires that cis interactions are stronger than trans interactions. Finally, I have shown that this patterning is related to cell fate, but it's not only early markers of cell fate, but is also sufficient to determine cell fate. So, well, finally, I would like to tell you a little about the future perspectives. So until now, I have been focusing on how notch signaling determines patterning, and it seems that now quite clear that now notch through lateral inhibition can generate these kind of patterns. But the question now is, what is downstream of notch that determines the generation of the final shape of the notochord? So which are the genes that are downstream of notch? But also, this is really a hugely mechanical process, and which are the forces involved in this kind of morphogenesis of the notochord? So, well, finally, I would like to acknowledge my postdoctoral lab, where I did this work, the Alvarez Muñoz lab at the EMBL. And now I would also, I am in a preparation to group leader position at the Karlsruhe Institute of Technology, part of the JICPrep Pro. And I would like to thank also Lisa Kretna, Marina Kanmaneva, and Magdalena, who are helping me to decipher how notch signaling is translated into different shapes. Finally, I would like to acknowledge different people who helped me at different stages of this project, discussing ideas or supporting me. So thank you very much. And I'm happy to hear and to listen any questions and to answer any questions.
Thank you, Hector. You already have a question.
Yep.
From Ruth Williams. Hi, Hector, very beautiful work. I was wondering if you looked for any evidence of oscillating gene expression.
So I haven't looked at it, but it could be very interesting. So now I have asked for a line that is a HER6 reporter, like it's really like the endogenous, like really following the endogenous dynamics of HER6. And I expect that this reporter will allow me to see if there are some oscillations. But currently the reporters that I have until now, they are stable. So they are kind of like integrating all the signal over time.
Okay, you have one more question from RT Criswell. Great talk. This might be a technical question. How did you mosaically label the notochord cells with jagged one to distinguish between cis inhibition and trans activation?
So I'm not sure if I got the question really well. So to mosaically label the cells, what I do is I have an amylin promoter that is quite specific for the notochord. And then I just inject in F0. And then I, just by chance, by randomness, some cells are, the transgene is expressed in some cells, but not in others. So that's the main,
Okay, another question from Joey Gersey. Hi, amazing work. I was wondering if you also tried to make jagged 1B reporter line. Maybe I missed it. What is triggering the expression of jagged 1A?
So what's triggering the expression of jagged 1A? I don't really know. There should be some general notochord developmental program that promotes all the cells to express jagged 1A. And then the jagged 1A is repressed in HER6 and HER9 cells. And then about the jagged 1B reporter is something that I tried and it's not yet generated. But what I can tell you is that it's interesting that the pattern of jagged 1B is quite similar to jagged 1A, but it's a little more diffuse. So it's something like, in the end, the pattern is the same when things are fully established, but jagged 1B takes more time to get this pattern. So it's more like homogeneous until it's really defined.
Okay, another question from Augusta Ortega. Very interesting talk. I'm curious if there's a dominant negative receptor mutation that you could express on the same cells that's expressing the ligand. Could this be a way to inhibit the cis effects of jagged 1 and not the trans signaling?
So I think there is people trying to find ways of distinguishing cis and trans, but the problem is that in general, jagged 1 and the receptor interact both in cis and trans using the same domains. So it's probably very difficult to find a way of specifically block cis, but not trans interactions. But yeah, it could be really cool and really something like to prove the hypothesis that then there wouldn't be any pattern. So it's really, very good idea.
Okay, and I have one question, and I'm not sure, I'm sorry if you mentioned it early, but which Notch receptor do you think is mediating these, this interaction in the cell fate choice?
Okay, so then, I think it's quite clear. So looking at the single cell RNA-seq, well, of course, I didn't show this data, but in the single cell RNA-seq, I could see that NOTS2 is by far the most highly expressed. And I could also confirm it at the level of mRNA staining. And also, it's not surprising that this is the one, because it's the one that is mutated in the Alakiel syndrome in humans and leads to vertebrae malformation. Okay. So everything fits, yeah. Okay. But I haven't really tried to mutate or to do to play with it.
Okay, all right. If there are not any more questions, I would like to thank Hector for a really great talk. And I want to thank Margherita and Hector both for their excellent talks. And I just want to remind everyone that the seminar is recorded and it will be available on the SDB website starting Monday. And now I really want to thank all of the postdocs who presented and moderated the Ethel Browne Harvey Postdoctoral Seminar Series this past year. So here in this slide, you can see all of our excellent postdocs who had a chance to share their work with the SDB community. And I really enjoyed watching all of this work and to actually see my fellow postdocs who conducted this work and how amazing and the variety of work that we have within the SDB community. I would also like to share with everyone that we will be opening the applications for the next set of presenters in June, so please be on the lookout for that. Any postdoc who is an SDB member is eligible to apply. So I want to thank everyone again for their amazing talks and a great seminar series, and we will see you back here in September.